Okay, it's 2:45, so we're ready to get started talking about zebra mussels. And I'm Kathy Erickson, the moderator for this session. I have a couple of reminders for participants. You've probably heard this a few times already today. But first of all, this session is being recorded, and you'll get notified after the meeting about how you can access the recordings. Secondly, if you have questions, the way you would enter those is using the chat box towards the center of the bottom of your screen. Just type in your question and I will be watching. If it seems like something that's kind of broader or an expansion, I might save it for the end, and otherwise I'll let the speaker know that we have a question and I'll relay it to them. So our first speaker in this session is Mike Schollmeyer. Mike is the secretary of the Mackenzie Lakes Association. Zebra mussels were first discovered in Big Mackenzie Lake, which is in Burnett and Washburn counties. They were on a dock found in the fall of 2016. Starting in 2017, MLA started an intense monitoring program using plate samplers in all three of the Mackenzie Lakes chain. Dr. Schollmeyer will present the results of 3 years of plate monitoring. So go ahead, Mike.
All right, thank you, Kathy. Next slide. Can we have the next slide, please? Zebra mussels, just for a short introduction here, were first found in the Great Lakes and Lake St. Clair, which is part of the Great Lakes system, in 1988, and in 1989, they were then found in inland waters in Wisconsin. In 1991, they were found in the Mississippi River and Lake Pepin, and by 2017, we've identified 306 locations found in 54 counties within Wisconsin. All these locations have been observed, verified, or non-verified, depending upon what was presented to the DNR. Next slide. As Kathy had mentioned, zebra mussels were found in Big Mackenzie on a dock when it was pulled in the fall of 2016. The shore owner was astute enough to take the mussel that they found and had it verified by the DNR at the Spooner office. Next slide. Mackenzie Lakes are northwest of Spooner, as you see here. It consists of a chain of lakes of Big Mackenzie, Middle Mackenzie, and Lower Mackenzie. They're all connected by Mackenzie Creek. Next slide. And they flow from Big Mackenzie, Middle Mackenzie, Lower Mackenzie, as I said, through the Mackenzie Creek. Big and Middle Mackenzie are actually in Burnett and Washburn counties, whereas Lower Mackenzie is in Washburn County. Next slide. In the winter of 2016 and the spring 2017, a zebra mussel team was composed of federal, state, local, county, and Mackenzie Lake associations to provide a risk assessment looking at how to prevent the spread of the zebra mussels, and of course the monitoring and education of the public, as well as making sure that we are following what is actually happening in the Mackenzie Lake chain. Next slide. Our biggest effort was in the monitoring of the zebra mussels. We did this by deploying plate samplers. Plate samplers are made of 4 plates of equal size, separated by 1 inch, providing 2 square foot of surface area, and they were actually placed in shady spots at the end of docks. Next slide. This is a picture of the typical plate sampler that we were using. As I said, we had People place these at the dock, usually in a shaded area between usually a boat and the dock itself. Next slide. We placed 24 of these plates in Big Mackenzie. We had 11 deployed in Middle and 4 in Lower Mackenzie. Next slide. Here's a representation of the actual addresses. We have deployed these in Big Mackenzie. As you can see, we were well deployed throughout the lake. Next slide. In Middle Mackenzie, we had 11. If you notice at the bottom of the screen on Big Mackenzie, you can see the Mackenzie Creek flowing from the north end to the south end of Middle Mackenzie. And on the west side, where you see a number of the deployments, that is a public landing. And then on the northeast side, you'll see where the outflow is from Middle Mackenzie, and that goes down to Lower Mackenzie. Next slide. Here's Lower Mackenzie. As you can see, the middle are the Mackenzie Creek coming in. And then on the north shore, you'll see the outflow where the creek actually goes into Namakagon, which eventually flows into the St. Croix River. Next slide. Here are the results of the first year of our plate samplers. In Big Mackenzie, we had 24 plates deployed. However, we lost 2 of them. One was stolen and one we lost in a windstorm. In that year, we had an average of 21 zebra mussels on a plate, or approximately 10 per square foot. On Middle Mackenzie, we only found 2 adults. One was on a plate, and we found one right next to a dock where that plate was deployed, actually on the wheel of a roll-in dock. In Lower Mackenzie, we found none. Next slide. In 2018, we had 24 plates deployed. 20 of those plates all tested positive for the zebra mussels. We had 4 of the plates non-reporting because either the owners moved or we just had no one reporting the results. Our range from the plates was from 5 zebra mussels to 473 with an average of 113, giving us a density of 56 per square foot as opposed to 10 per square foot from the previous year. On Middle Mackenzie, we had 3 of the 11 plates were positive, but only a total of 5 zebra mussels found. In Lower Mackenzie, we had no zebra mussels found. We also had 2 sentinel lakes. One was a completely private lake of approximately 54 acres. We deployed 1 plate there, and that turned up negative. And then we had another sentinel lake with no public access, however, people could access it from the road. That was a 17-acre lake, and that was also negative. Next. In 2019, after we pulled the plates on Labor Day, we had, of the 24 plates, we had 6 that were non-reporting. However, the plates were so inundated with the zebra mussels, they were actually too numerous to count, and I'll show you a picture later on of why that was the case. In Middle Mackenzie, we had 11 plates. All were positive for zebra mussels with a range of 18 to 248 of the adults, with a total of 911 zebra mussels found on those 11 plates, with an average of 91 plates or 45 per square foot, a tremendous increase from the year before. Lower Mackenzie, we have still not found any adults on any of the plates or on any docks reported. Next slide. Here's one of the plates. Typically what you see, I would say this is a plate that didn't have nearly as many as some of the other plates do, but you can see on that top plate that we have zebra mussels on top of zebra mussels on top of zebra mussels. Next slide. This is a boat lift that was pulled out of Big Mackenzie. As you can see, the tremendous number of zebra mussels that we had. I believe this is a wheel. Next slide. Here's the end of the lift. As you can see, all the zebra mussels on the crossbars. Next slide. For 2020 monitoring plan, we have, as I mentioned before, we were monitoring this through the Rapid Response Grant. That grant has now been completed and it lasted for 3 years. Even though we do not have a grant, we are still going to continue to monitor. We're going to put out plate sampling in all 3 lakes, but we'll concentrate on middle and lower because these are the lakes that we are trying to get a more accurate representation of what's going on, whether we're going to see a continual increase or whether we're going to see any in the lower Mackenzie. Lower Mackenzie is fairly unique in that its calcium content is lower than the other 2 lakes, and so we're wondering whether there will be enough calcium in the water to actually help adults survive. And if we'll hopefully give you another update by next year. That's the end of my presentations. If anybody have any questions.
Thank you very much, Mike. I wish the news was better, but I'm glad you're watching. I don't see any questions at this point.
Okay, thank you.
So our second speaker on the zebra mussel topic is Angelique Dahlberg. She's a student at the University of Minnesota in the Conservation Sciences program. Today, Angelique will discuss results from a 2019 project investigating the effectiveness of EarthTech QZ on controlling zebra mussel veligers in Lake Minnetonka. She'll share research plans for 2020 and beyond. The project is a collaboration between the Minnesota Aquatic Invasive Species Research Center and the U.S. Geological Survey. Angelique received her ES in Biological Aspects of Conservation from the University of Wisconsin-Madison and her MS in Integrated Biosciences from the University of Minnesota Duluth. Additionally, she has worked for the St. Croix River Association from 2014 to 2018, and prior to that for the Cook County Invasives team. And I'll just mention that when she worked with St. Croix River Association, she also helped put on a conference, which we greatly appreciate. So Angelique, go ahead, please.
Thanks, Kathy. Can you hear me okay?
I can hear you.
Okay, great. Before I got started.
If anyone else can't hear, please send us a chat.
Great. Well, good afternoon, everyone. As Kathy said, my name is Angelique Dahlberg, and I'm going to present some of the ongoing and future research for zebra mussel suppression happening here at the Minnesota Aquatic Invasive Species Research Center. Next slide. Thank you. So first of all, a little background on our center. The Minnesota Aquatic Invasive Species Research Center, which we shorten to the acronym MERC, has a mission to develop research-based solutions that can reduce the impacts of aquatic invasive species in Minnesota. We do this by preventing spread, controlling populations, and managing ecosystems. And ultimately, our goal is to advance knowledge to inspire action by others. Next slide. So one of the species that we target are zebra mussels. Mike did a great job introducing zebra mussels, so I don't have much to add. To the left on this slide is an enlarged photo of a zebra mussel, and to the right is a map of which lakes and rivers in Wisconsin have confirmed zebra mussel populations. as of March of this year, there were a total of 252 lakes and rivers that fell into that category. Next slide. Next slide, please.
I'm trying.
Thank you.
Not sure what's going on here.
Thank you. Here at MERC, we have 6 current projects that focus on zebra mussels and 14 completed projects. To learn more, you can visit our website, which I'll have written out in a few slides. That's www.merc.umn.edu. And in the next couple of slides, I will touch on the main areas of our zebra mussel research. After that, I'll get into the research that I'm involved with. Next. Some of our projects work to better understand the pathways of zebra mussel spread. For example, past projects have included identifying the genetic relationship between lakes, performing risk analysis on residual water in boats, and developing network models to predict invasions. Next. Other projects work to improve zebra mussel detection. Our researchers have evaluated survey strategies, written survey protocols, developed sensitive environmental DNA detection assays, and tested sonar and sensor detection methods. Next. Still, other projects, including the one that I'm going to talk about, examine ways to control zebra mussel populations. Next. Finally, our researchers also work to improve efficient management. Research in this area has included optimizing decision tools for locating watercraft inspectors and creating models to simulate management scenarios. Next. So I recognize that that was a very brief overview of our zebra mussel research at MERC. If you want to learn more or connect with us, you can visit our website, which is on here, or connect with us on social media. Additionally, we have an annual research showcase every September. This year's showcase is scheduled for September 22nd. Traditionally, it's been held in person. There have been no decisions about how that will be held this year, but we will likely have some content presented at that time in some format. Next, please. So now on to the project that I've been involved with. This research is investigating copper-based control of zebra mussel settlement and the associated non-target impacts that the treatment may have. So to start off, I want to quickly acknowledge our funders and collaborators in the project. And I think this is very important. I know we have a broad audience today, so I wanted just to mention these up front because we have received a variety of support from many levels, from the lake level to the county level to national government agency level. So our funders have included the Minnesota Environment and Natural Resources Trust Fund, the Bay Lake Improvement Association, the Pelican Lakes Association of Crow Wing County, Hennepin County, the Great Lakes Restoration Initiative, the Fletcher Family Foundation, the USGS, and MERC. So it's been truly a collaborative effort to make this happen. So why copper? Next slide. Well, we've been using copper for aquatic management for a very long time. Historically, copper plating was used on boats as an anti-fouling agent to prevent the attachment of barnacles. And some records suggest that we were plating our boats with copper as far back as ancient Egyptian times. More recently, beginning over just about the past century, researchers and communities began using copper sulfate to control algae in lakes. Originally, they would go out in boats and have burlap sacks of copper sulfate crystals and drag them through the water for treatment. Since then, we've continued to use copper sulfate as an aquatic algicide, herbicide, molluscicide, and for macroinvertebrate control. And today we've extended those uses to include control of aquatic invasive species, including zebra mussels. And this use for zebra mussels isn't new. Back in 2011, Rose Lake and Lake Irene in Minnesota both used copper sulfate for adult zebra mussel control. However, we do still have a lot to learn in this field, and so that's where our project comes in. Next. Thank you. At MERC, our research started with trials in the lab and then scaled up to test plots in the lake. We found that copper products are toxic to zebra mussels and that veligers, the larval life stage of zebra mussels, are more susceptible than adults to lower concentrations. One report that we came out with, I think it was published last year, gives recommendations for adult zebra mussel control, including copper sulfate use. And that report is available on our website at that link at the bottom. And so if zebra mussel control is something that you're interested in, I encourage you to check that out. Thank you. Next. So our research projects for this project, which is building on all of that research I just talked about, our research questions are, can we prevent veliger settling and or control veligers with a low dose of copper, understanding that veligers are more susceptible than adults? Is it possible to treat only the top layer of the lake or the epilimnion and have effective treatment? And does a low dose of copper decrease ecological risks, especially risks to non-target organisms? Next. So in 2019, our team, composed of researchers from MERC and from the USGS, tested a low dose of copper on St. Albans Bay and Lake Minnetonka. Lake Minnetonka, just to place you, is in the western part of the Twin Cities metropolitan area. It's had zebra mussels since 2010, and the complexity of the lake and the different bays made it a sort of intuitive first lake to use for this purposes. Our control bay and our test bay were very separate from one another. You'll notice on this slide that our control bay, which is Robinson Bay up there in the northeast corner, I've abbreviated to ROB. I'll be using this notation throughout the slides, so I just wanted to point that out right now. That bay was about 37 hectares with a maximum depth of 19.1 meters. St. Albans Bay, which is the bay that we treated with copper sulfate, I've abbreviated to SAB, and it was 66.3 hectares with a maximum depth of 11.3 meters. Next. So at both sites or bays in this lake, we have 5 plots. And so I have a schematic of how this worked in like a cartoon format on the left. And then St. Albans Bay, which was our treatment bay, there's an aerial image over on the right with some of the bathymetry. So we had 2 bays. Within each bay, we had 5 plots, which were marked by buoys. And at each plot or buoy, we monitored zebra mussels and non-target organisms. And we did that through veliger tows, use of sampling plates. We had caged fish, caged zebra mussels and native mussels. We did zooplankton tows, benthic grabs, and we took water samples. And I'll go into more detail on all of that, but just to give you a broad view of what our study design looked like. Next. The treatment itself took place over 10 days. We started it last July 20th, and we used a total of 1,650 gallons of EarthTech QZ over that duration. Our first treatment was at a dose rate of 100 micrograms of copper per liter. We then conducted bump treatments to maintain an elevated copper concentration on odd numbered days, starting on day 3. So on this figure, the star denotes the initial dose, and the plus signs are the days that we did the bump treatments. And we did the bump treatments at 60 micrograms of copper per liter. And so our goal was to maintain that concentration of at least 60 micrograms of copper per liter, which is denoted here by the dashed line. We monitored our concentrations in the lake in 2 ways. One was done in the field to allow for day-to-day decision-making. And then we validated that later in the lab. So we also collected samples, sent them to the lab, and we wanted to make sure that all of the things that we were doing in the field were what we thought we were doing. And that generally is true. There's a little bit of difference between our 2 methods here. You can see with the difference in the lines, but in general, we were doing what we thought we were doing, which is always good, always what we want. So we paid attention to, we were able to closely track the amount of copper that we were adding to St. Albans Bay. Next. And this is a similar figure. When we look at the copper concentration in the treated bay, which is the black here, and compare that to the control bay in red, we can easily see that we did in fact have elevated levels of copper in comparison. We also, and this is perhaps even more interesting, you can see that on July 21st, at the beginning of the treatment, our copper concentrations are very similar. And by early September, certainly by November, our copper concentrations in the treated bay are back down to that level. So we know that within a matter of just a few months, all of this copper is no longer seen in the water column. Next. And this treatment, of course, was to control zebra mussel veligers. So we conducted veliger tows immediately before treatment, 1 day following our first day of treatment, and then 14 days after treatment concluded. And in this figure, we're looking at the number of veligers per liter of water for all 3 tow dates for both bays. And what we saw was that treatments effectively reduced zebra mussel veliger density, and apparently quite quickly, which is what we were going for. So that's a promising preliminary result there. Next. Our settlement plates tell us basically the same thing. We placed settlement plates in both bays, and we retrieved some of those plates 30 days after treatment and the rest 90 days after treatment. So here the light blue is the control bay, the dark blue, which you don't see at all, is the treated bay. So you can see that there is substantial attachment in the control bay, but there is essentially no attachment in the treated bay. So the veligers are disappearing from the water column, and they're not appearing on our settlement plates. Next. In addition to impacts to zebra mussel settlement, we also looked at a variety of non-target impacts. First among those, we monitored water quality. And as we expected, we found no changes to water quality due to treatment. On the right are results for pH and dissolved oxygen. You'll see that for both bays during the treatment period and after the treatment period, there's a little bit of variation between the 2 bays. They were, I measured it at one point, I think they were about 5 water miles apart. So they're quite a ways apart, but there's really no change due to treatment there. We also monitored temperature, specific conductance, chloride, dissolved organic carbon, sulfate, calcium, magnesium, sodium, and potassium. Next. We did find some impacts to algal productivity as measured by chlorophyll A. The relationship with algae is a little interesting. Copper sulfate has, as I mentioned earlier, been used as an algicide, and it still is pretty commonly used as an algicide. So we know that generally a product like this will have an impact on algae. We see that pretreatment levels are very similar in the two bays, and then the productivity increases in the treatment bay, and then finally decreases, while the control bay stays relatively similar with a little bit of increase as we go into August. One possible explanation for the treatment bay is that because copper is an essential micronutrient for plants, including algae, it's possible that that initial addition of copper spurred a slight growth. And then as the copper concentration crossed a threshold, it may have acted more as an algicide, but more research is needed to better understand this. Next. We found a decrease in the abundance of benthic invertebrates and benthic invertebrate family richness. We monitored benthic invertebrates before treatment, 1 day after initial treatment, and then 14 days after conclusion of treatment. And both the number of individuals and the number of families represented declined. And I've presented that here visually, but I do want to point out that the bays started with very different numbers, especially if you look at the figure on the left with the number of benthic invertebrates. You'll see that even in the pre-treatment, the control bay has quite a few more than the treatment bay. So inevitably there were pre-existing differences between the bays. But in general, we did see a decline in the treated bay. Next. We saw similar trends in the abundance and family richness of zooplankton. Again, we monitored zooplankton the same way we did for the benthic inverts, before treatment, 1 day after initial treatment, and 14 days after conclusion of treatment. And we saw both the number of individuals and number of families represented decline. Next. In addition to the organisms that we monitored that were already living in the lake, the algae, the benthic inverts, the zooplankton, we also monitored caged fish, caged native mussels, and caged adult zebra mussels. We placed fat mucket mussels, fathead minnows, bluegills, largemouth bass, yellow perch, and adult zebra mussels in independent cages beneath each buoy in both bays. So we had a bunch of cages beneath each buoy. The cages went in the day before treatment, and they were removed the day after treatment concluded. And so these results that I'm showing here are from when we removed the cages, and we're looking at percent survival. So when we took out the cages, we found that adult zebra mussels had lower survival in the treated bay than the untreated bay. We also observed that there may be a difference for fathead minnows, but more research is needed to understand that. There's a little bit of, we were not able to recover all of the fathead minnows, so it's a little unclear there. We need more samples. And the other species did not show decreased survival due to treatment. And again, as I say this, I want to draw your attention to the fact that it's not overall survival that we're looking at. So you may notice yellow perch has a low percent survival, but it has a low percent survival for both bays. And we suspect that's due to transportation to Lake Minnetonka initially, and they didn't acclimate as well as some of the other fish did. However, when we look at the zebra mussels on the flip side, we see that there is a dramatic difference between the treated and the untreated bays. Next. So once those fish and mussels were taken out of the lake.
Excuse me, Angelique. Just got a question about, could you explain what are benthic invertebrates?
Yeah, so the benthic invertebrates are some of the what's the best way to describe it? Like some of the small invertebrate animals that live in the sediment at the bottom of the lake. So like small, spineless, bug-like creatures. I don't know that that's like the most textbook definition, but.
And maybe there's a way to explain why it would be bad to lose them.
So we are, one of the things we're concerned about with this study, all of the different organisms in the lake have a different role in the food web, and when zebra mussels enter a system, they dramatically change that food web, and they outcompete some species, they out-eat other species. But when we treat a system with something like copper sulfate, we are also, there may be non-target impacts, and we may be playing with the food web in other ways. And so understanding which organisms or elements of the lake are affected will better help us understand what's happening and what some of those effects that we're not necessarily measuring might be.
Thank you.
I hope that was somewhat helpful. Okay, thank you. That was good. Yeah, thank you. So I forget where I was in here, but I guess with our caged animals, once we took them out of the lake, we sent them to the lab and analyzed their tissues for copper accumulation. And that's my friendly way of saying that they did not continue their lives after the study. And we found that the fathead minnows, fat mucket mussels, and zebra mussel adults had higher copper concentrations in their tissues following the treatment. So here, the untreated bay, the control bay, is Robinson Bay on the left. The treated bay is on the right, and the copper concentration in the tissues is shown. So you'll see the bars on the right are noticeably higher than the bars on the left, suggesting that there was some sort of accumulation in these animals. Next. So finally, we compared the resident or wild zebra mussels in the lake between the two bays before and after treatment. And just to place this like in the context of the study, we had our pre-treatment work that happened. We had a 10-day treatment window. We took the cages out right after treatment. We did some work about 2 weeks after, and now this is part of the longer term work in 2019 that we did. So we sent a scuba diver down to do transects in early July before the treatment, and then in September, about 2 months after the treatment. So we do have the survival rate that I just showed you on the previous slide for adult zebra mussels that were in the cages, but these scuba transects give us better insight into what happened in the several months following treatment. So we saw that there were more caged adults that died in the treated bay than the untreated bay. But when we sent our scuba diver down, we found that 2 months later, almost none of the resident zebra mussels were still alive in the bay, which suggests that in addition to killing the villagers, we also pretty effectively killed the adults, which is, you know, the intent of a zebra mussel suppression strategy. Next. So in summary, treatments effectively reduced zebra mussel veliger density, juvenile zebra mussel recruitment, and those adult zebra mussel densities in quadrat samples along the transects from the scuba diver. The non-target impacts to the things like the benthic invertebrates varied during the course of the study, and we don't have any definitive results that are really ready to be published yet. In general, we saw that the zooplankton decreased after exposure. We saw that benthic invertebrates decreased after exposure, but we will, and this is pretty key, we will conduct follow-up monitoring in 2020 and 2021 to look at what those numbers look like 1 and 2 years out, and to see if there was any rebound and what that might look like. Next. So moving forward from the work in Minnetonka, our next step is to try to improve our methods and work with a second lake. So we were targeting veligers and we had great success, but we did also see some mortality of adults. And if we know that veligers are more sensitive than adults, this suggests that maybe we can go even lower. So how low can we go and still suppress veliger settlement? And if we go lower, will we also further minimize our non-target impacts? So to answer these questions, we want to conduct the same type of study on a second lake. Next. We know that copper toxicity changes with water quality, and we know that we can predict copper toxicity using a tool called the biotic ligand model. I won't go into that a whole lot. It's really, I'll just mention it, but it's a model that's used by the U.S. Environmental Protection Agency to predict and determine copper toxicity levels in aquatic system, freshwater aquatic systems. So we want to figure out, if we collect water quality measurements on a lake, can we use this model to predict the lowest amount of copper to use to suppress veligers, and then test it and see if it's effective, and look at those non-target impacts and hopefully minimize those. Next. So as a sneak peek into our processes, we are currently selecting a second lake in Minnesota for treatment. We're working with the Minnesota DNR to identify an appropriate lake right now, and ideally we'll treat that lake in 2021. We're going to continue monitoring in Lake Minnetonka this year and next year, and potentially beyond that. We'll treat that second lake next year, and then following that treatment, we would continue monitoring that in 2022 and again, potentially beyond. No decisions have been made as to which lake we're going to treat for our next test. So I guess I'll just say stay tuned for further progress on that. Next. And
Question, Angelique? Oh,
okay, well, I was gonna say with that, I think I'm ready to take questions. So I'm gonna leave this slide up in the background. And if you have any questions that we don't get to today, or you just wanna reach out and talk more, my contact information is on here, as is my collaborators, Diane Waller with the USGS.
Okay, I don't know if you can see the questions, but Lisa Burns asked, are there any effects on aquatic plants from these treatments?
We did not look at aquatic plants for this. So we don't have any metrics on that. We did not observe any, but we weren't measuring that in Minnetonka.
And another question from Megan Williams. Thanks for the interesting presentation. Just wondering if you plan to measure copper in the sediments. Also, did you measure total and dissolved copper in the water column?
We have been talking about looking at dissolved copper in the sediment in our second lake. And I'm trying to think. I don't recall if we measured both in the water. I think we measured, I do think we measured both, but I'd need to confirm that. We were, there's a whole large team of us. And so I was not in charge of the copper concentration in the water part.
Okay. And I see that Patrick put Angelique's email address in the chat box, so anyone else who wants it can see it there.
Thank you.
Just chimed in with another question. How big of an area was tested and what was the treatment cost?
Let's see, let me pull that up. The St. Albans Bay was 66.3 hectares, and we treated only the epilimnion, or that top layer of the lake, and that in this bay was about 5 meters down. So I don't have the total volume, but 66.3 hectares in many areas down to about 5 meters. And we used a total of 1,650 gallons for a price of $22,000. But that was just for, we were able to get the product at retail cost. So, I mean, likely it would be much more in like a private lake scenario. And that does not factor in like applicator time or staff time for any monitoring, et cetera.
Can you give us a conversion from how many acres in a hectare?
Not offhand.
Not
Oh,
offhand.
thank you. Here I see
Patrick can.
Patrick.
2.4.
I know I was going to see if I could Google it quickly, and then I was like, no, just so you don't know.
That's what I did.
Okay. Any other questions? Type quickly if you do.
I know I like had a lot of information that I presented there. We're pretty excited about this project. So hopefully everyone had some take homes there.
Would you describe it as promising news?
I'm an optimist. Yes, I would.
Okay.
I mean, there's still, it's really hard. So it is a product that's registered for use, but it's also, we're still very early on in research stages. There's a lot that happens between like a very controlled research setting and like a successful everyday application. So, but I think it's really exciting that we're trying to find the lowest possible concentration that would have effective population suppression. Because so often, we treat to the labeled amount, but there are a lot of non-target impacts. And so I think that's very promising. I think it's a really good focus.
That's good. And there was another question that came in from Douglas Peterson. What about genome research?
Oh, great question. So the genome of the zebra mussel was announced and made public last summer. So that is available. We do not have a current project on that. I know MERC is sort of poised to take the next step. It it just hasn't quite fallen into place yet. But we are still very invested in that. And the genome is, if you visit MERC's website, there's more information about that and how to access it if you want that.
Okay, and then another question going back to the copper treatment. What is the potential for scaling up this copper treatment?
I think there's good potential. Part of that might be like more of a regulatory. There's a regulatory side to that, and how much the DNR might permit. We had very good luck treating only the epilimnia in that top layer, and so that means less product. And I mean, the bay itself was the size of a small lake. So I don't know. I mean, for what that's worth, I think certainly for smaller areas, it could easily be scaled up. I don't know about some of our larger lakes like Winnebago, but and then of course there's a price tag to everything.
Yeah.
So
Okay, it looks like that's the last question. We'll pause a few more seconds here. But thank you very much, Angelique, and to Mike too, and to all of our registrants who joined us today. This is the last session. We're glad you could be here, and we're looking forward to next year.